But before we can really talk about what the light-dependent reactions are, we have to talk about light energy. Light energy comes from electromagnetic radiation, which comes from the sun. This is a wide spectrum of wavelengths. The shortest wavelengths are the gamma rays to the longest wavelengths of radio waves. This slide, if it's not review, you should probably just leave. Humans can only see a small portion of the spectrum. We call that visible light. Energy travels in waves. The top of the wave is the wave, or the crest. The bottom of the wave is the trough. From crest to crest is what is measured as wavelength. Is that, yes? Yes. Wonderful. So, this is all put together. We call it the electromagnetic spectrum. This is the range of all possible frequencies of radiation. Each type of radiation travels at a certain wavelength. If we have very long wavelengths, this would be like your radio waves. They carry less energy. In your shortest wavelengths, like your gamma rays, carry the most amount of energy. The higher the energy waves are, the more likely that they can penetrate tissues and damage cells and DNA. So, like we know X-rays can penetrate human tissue, but not bone. That's why we get the image of a broken bone when you go in for an X-ray. So, everything has pigments really, have the ability to absorb light. We are going to be focusing on organic pigments because we're dealing with plants and plants are organic. They have a narrow range of energy levels that they can absorb. That means they don't absorb the whole spectrum of energy. The energy levels lower than red light are not sufficient and they don't have enough energy to raise an orbital electron to an excited state. So, quick chemistry refreshers. We have our nucleus surrounded by electrons that are orbiting. Yes, remember this? So, there are 7 potential orbits. And if this electron gets excited because energy hits it, it will bump up to the next energy level. So red light doesn't have enough energy to push that outermost electron to the next energy level. If we have blue light has plenty of energy to do this. However, it has too much energy. Yes, your poor eyes are getting schmucked up because of the blue light from your phones. Yes. So the blue light will physically tear the molecules in those pigments apart, and then those pigments cannot function as they are intended to do so. This process is called bleaching, which kind of makes sense. If you bleach something, you turn it white. Okay. Your human eye, your retinal pigments and plant pigments have the ability to absorb wavelengths between 700 nanometers and 400 nanometers. Those happen to be the wavelengths of the rainbow of colors. When we talk about in terms of plants, this is called the photosynthetically active radiation, meaning it works for photosynthesis. Okay? So we have to understand how these pigments can work before we can really talk about photosynthesis. So first of all, different kinds of pigments can absorb only specific wavelengths of visible light. Pigments then reflect the wavelengths that they cannot absorb. So we know chlorophyll is what color? Green. That means chlorophyll doesn't actually absorb green light. It reflects it back. Okay, so like Sid's sweatshirt is red, the pigments in those dyes are absorbing every other color except red, and red light is being reflected back to our eyes. So the color you see is the color that is not getting absorbed. It is being reflected. Yeah, we'll just let that sink in here for a little bit. All right, so there are two major classes of photosynthetic pigments that are found in plants and algae. The first one you're familiar with, chlorophyll. We've been preaching chlorophyll since 7th grade. However, there is a second type of pigment found in plants called carotenoids that also help with photosynthesis. So back to chlorophylls. There are 5 major types of chlorophylls. Chlorophyll A, B, C, and D. And then there is a special bacterial chlorophyll found in bacteria. Good job. So chlorophyll A is a form of chlorophyll that absorbs violet, blue, and red light. And it will consequently have a bluish green color to it because the greens are not getting absorbed, so they're getting reflected out. This is the only pigment molecule that performs photochemistry by getting excited and losing an electron to the electron transport chain. Chlorophyll B is an accessory pigment. That means it's an extra one. It's not the main one. That's why it's the B, not the A. And it absorbs blue and reddish orangish light, and it gives a yellow-green tint. The carotenoids, these are photosynthetic pigments that range from yellow, orange, and red, and they actually have a very important job. They dispose of excess energy. So think of a leaf. A leaf is taking in a crap ton of energy, more than it needs to do photosynthesis. And so extra energy can be dangerous because it can tear things apart and wreck them. And the carotenoids absorb that extra energy and turn it into heat energy, and the plant then releases a little bit of heat. Let's see, these are located in the thylakoid membranes, and this is actually a bigger group of pigments than chlorophylls. They're found in fruits. You've probably actually heard of some of these, like the red in the tomatoes is called lycopene. The yellow of corn is called zeaxanthin. And oranges that make them orange, the orange peeling, is full of beta carotene. I feel like those words come up on like vitamin commercials all the time. Chloe, you're just saying yes, but you don't really mean it.
Is that why carrots are orange too?
Yes.
Because of carrots?
Yes. Good job, Brett. All right, so we need to understand these pigments. So remember, each pigment only absorbs a certain range of wavelengths. And each pigment has its own absorption spectrum. And this is the range of wavelengths of electromagnetic radiation absorbed by a given substance. We use this to identify the pigments. The device we use to create an absorption spectrum is called a spectrophotometer. And this is an instrument that measures transmitted light and computes the absorption. So they're really they're probably about a box like the size square of my podium up here. And you put the liquid color of whatever, so like you can extract chlorophyll from a plant and you can put it in this tiny, it almost looks like a test tube, but it's more square. And it's called a cuvette. You put it in, then you hit the go button. The go button shines a light through it, and then it spits out a report at the end and tells you what colors of wavelength were absorbed and which colors were not.
That's cool.
Yeah, it's super cool, super easy to use. I've never used one until I went to college. And you can do this with other pigments. They don't have to be just like chlorophyll or the carotenoids. They can be like dyes and like for paints and stuff too. So the next slide shows you an absorption spectrum for chlorophyll A, B, and the carotenoids. Each peak will indicate what wavelengths are absorbed. So if you look here, on the left-hand side, these are your chemical structures of, this is chlorophyll A. This one's chloro B, and this is carotenoid. If you look, these red boxes are indicating to you the only molecular difference between chlorophyll A and chlorophyll B. But the important part of this absorption spectrum is the image here on the right. The solid line is for chlorophyll A, and so it's telling you that chlorophyll A really absorbs here, here, in those wavelengths. And so you could go down here. Ooh, what wavelength is this? 675-ish? Yes, yes? And then what you notice is down here, it's not absorbing any of those wavelengths. So that tells you, if you want to know what color you're going to see, you look for the trough, the valley, where it's at 0 on that x-axis. Yes? Okay. So the longer dashes, this one right here is for chlorophyll B, and lastly, the dots are the beta-carotene. Are there questions on this absorption spectrum?
What's the third one down?
The beta-carotene for like the carrots.
Okay.
All right, so now we finally get to look at how the light-dependent reactions work. So overall, their goal is to take solar energy and convert it into chemical energy in the form of NADPH and ATP. That chemical energy then supports the light-independent reactions and fuels the assembly of sugar molecules. So in order to do this, we use protein complexes and pigments, and they work together to produce those energy molecules, the NADPH and the ATP. And so this occurs in what we call two photosystems. What does the prefix photo mean again? Light. That's a thing to keep in the back of your head as we go through this. This first one up top here is called photosystem 2, which will get abbreviated PS Roman numeral 2. This one is photosystem 1, and it will get abbreviated PS1. And they do go in this order. It starts with photosystem 2 and then goes to photosystem 1. However, photosystem 1 was discovered before photosystem 2, hence the flip-flop of the 1 and the 2. Okay? Yes, 2 to 1. Just count backwards. Okay? So each photosystem is a group of proteins, chlorophyll, and other pigments that are used in the light-dependent reactions to absorb light and energy and converts it into chemical energy. Photosystem 1, the integral pigment and protein complex, is located in the thylakoid membrane. It uses light energy to transport electrons from plastocyan to NAD+ or NADP+, which becomes reduced to NADPH in this process. Photosystem 2 is another integral protein and pigment complex located in the thylakoid membrane that transports electrons from water to the electron transport chain. And this important part here is that oxygen is a product. So remember, what do we need for photosynthesis? You need carbon dioxide and you need water. So in photosystem 2 is where that water gets used. And then what is a product of photosynthesis? Oxygen. So in photosystem 2 is where that excess oxygen gets produced and then released back into the environment. So your photosystem structure, this doesn't really matter. This is about the same for 1 and 2 here. They both have antenna proteins. They both have a reaction center. They both have light harvesting complexes, and they both deal with photons. So the light harvesting complex is this lighter green material, not the circles within it, but the lighter green materials on either side of it. And this is the complex that passes energy from sunlight to the reaction center in each photosystem. Your reaction center is this blue center area. Your antenna proteins are the pigment molecules, these guys, that they absorb light and transfer the energy absorbed to other pigment molecules. So it's kind of provides that hopscotch path for the light to follow. So you can see here that the light is entering and it hits one of the antenna proteins or the pigments, and that photon of light energy bounces from pigment to pigment until it reaches two pigments in that reaction center. Okay? This takes about a millionth of a second. So it's super fast.
Science is weird.
Yes.
Oh my God.
Yes, it is.
Wait, if it
All right, so, why didn't it change? All right, so this might work best if you look at Photosystem 2's picture, but we kind of talked about this already. So a single photon of light is going to be absorbed by a chlorophyll, so that would be one of the circles in the reaction center, or not the reaction center, but the complex. Like
the dark green one?
Yes. And this causes that chlorophyll to become excited. The energy then is then transferred from chlorophyll to chlorophyll until it is delivered to the reaction center. Only the energy is being transferred. It's not losing electrons, it's just energy. The reaction center then contains a pair of chlorophyll A molecules, which were like the two little ones at the bottom. And those actually have a special property. They can undergo oxidation upon excitation. So once they get excited with that energy that's hopscotched from chlorophyll to chlorophyll to chlorophyll, that energy then excites those reaction center chlorophyll A's, which causes photoact to happen. Photoact is the ejection of an electron from the reaction center using the energy of an absorbed photon. So this light energy is then converted into an excited electron. So basically, we're doing our hop, skip, and jumping. We get to the two special chlorophyll A's, and then you can see that there's that electron in the reaction center that gets shot off, and it moves up to this, what we call a primary electron acceptor, right up here. Okay, so that electron then gets connected to the primary electron acceptor.
So is the reaction center the blue part?
Yes.
Okay.
Yes. All right. So all of the following, from now on, all of the following steps involve getting that electron onto the energy carrier NADPH for delivery into the Kelvin cycle, which is part of the dark reactions. So photosystem 2 really is the photosystem that takes light energy and is converting it, and we extract an electron. After photosystem 2, we talk about electrons and not energy. Yes? Okay. So, both photosystem 2 and photosystem 1 have photosynthetic electron transport chains. This includes our friend Site C from cellular respiration electron transport chain. So this visual gives you a lot of information. Here is our PS2. Here's our first electron transport chain. Then we have PS1, followed by our second electron transport chain. And then, anybody remember what this guy at the bottom is? Yes, ATP synthase. Which does what? Makes ATP, very good. So this first electron transport chain takes the excited electron from PS2, which is right here, and hooks it up with this carrier posquinone, we'll just call it PQ. PQ shuttles the electron to site C. Site C shuttles that electron to PC, which is actually a protein called plastocyanin. And then finally that electron via PC gets moved into photosystem 1. Okay? So also an important thing to notice is part of the electron transport chain is the movement of hydrogen ions, because we're also creating a hydrogen ion gradient, which then we know in turn powers this process down here. Okay? So, photosystem 2. Its reaction center, so that blue piece of the diagram, is called P680. And it delivers high energy electrons to the primary electron acceptor and then moves the electron through the electron transport chain to PS1. So your primary electron acceptor is usually a pigment or another organic molecule in the reaction center that accepts an energized electron from the reaction center. P680's missing electron will then get replaced by extracting a low energy electron from water. This is the water splitting stage of photosynthesis. So let's just go back here. This diagram will work. So this electron that ends up leaving, it has to get replaced so an electron can leave again, right? And that comes from this process right here of splitting a water molecule. Okay.
So does the others, so it just like, so it like leaves and then the other stuff?
What is it? The electron?
Yeah.
Yes.
So then the other stuff is just like floating there?
Like the hydrogen and the oxygen. The hydrogen ions actually are some of the ions that will get pumped into and out of the thylakoid lumen. And then the oxygen gets released as a waste product of photosynthesis. We like that. Let's see. PS2 is re-reduced after every photoact. So after every time we lose that electron to the electron acceptor, we split that water molecule and add another electron to the reaction center, reducing it. Splitting, one water molecule releases 2 electrons, 2 hydrogen atoms, and 1 oxygen atom. However, you do have to split 2 water molecules because a single oxygen atom is not stable. It's actually toxic. LE or EL? Okay. So we have to split 2 so the O is not uncombined. And those of us that took chemistry, we know this makes sense because oxygen has to be a diatomic. Okay. So, as that electron moves from PS2 through that electron transport chain all the way to PS1, it starts to lose energy. That energy is used or lost to the movement of those hydrogen atoms from the stroma to the thylakoid membrane. Those hydrogen atoms are plus the ones produced from splitting the water, accumulate in the thylakoid lumen and will be used for ATP synthase. The electrons must be re-energized by PS1, and this is where we absorb another photon of energy by the PS1 antenna. Do I have a picture of it? Yeah. So here is that second absorption of a photon, and here's that electron that's moving in, and notice how they do meet up to re-energize that electron. Let's see, energy is relayed to the PS1 reaction center, which is called P70. It becomes oxidized and sends high energy electrons to an NAD plus to form NADPH. Then we produce NADPH roughly at the same rate as we produce ATP. So the electron moves to the 2 chlorophyll A's, just like in PS2, and it energizes or connects with the electron carrier up here, and that moves then to a carrier protein, slides it to the NADPH reductase, which then makes NADPH plus a hydrogen ion to make NADPH, which is a goal product of photosystem or of the light reactions. And then we've created a concentration gradient of hydrogen ions, which then allows us to make our ATP via ATP synthase in chemoosmosis. Basically the same process that we talked about last chapter.